Hi, I'm Martin Jankowiak, and it's my pleasure today to tell you about recent joint work that was done at the Broad Institute along with Fritz Obermeier and Jacob Lemieux. So the context for today's talk is the COVID-19 pandemic, and specifically the fact that there's been this huge global effort to collect a lot of genomic samples from COVID-19 patients. And so as of a few days ago, we've now collected something like 13 million viral genomes of SARS CoV-2. And so this is really a huge amount of data. It's a very rich data source. And we've never done anything like this, right? No one's ever sequenced this many viral genomes, and we've never seen a virus evolve in real time like this. So from a scientific point of view, this is all very exciting. And given this kind of rich data source, there are a number of more or less ambitious questions we might try to ask and answer, including the following questions. So we might ask, which mutations are driving the pandemic? Which variants or lineages are the most fit? Which newly emergent variants should we be paying extra close attention to? Which mutations are implicated in immune escape? Is there any evidence for epistasis or interaction effects between different mutations? And so on and so forth. So before we jump into trying to see how we might answer these questions, let's take a closer look at the data. So there are a number of ways to look at this data, right? So what do we have, right? We have millions of genomic samples, and we also know where the sample was taken from, let's say France or New Jersey or what have you. And we also know when the sample was taken. And so there are a lot of ways to slice and dice this data. And one useful way is, as is done here, using this visualization from NextStrain. So across the horizontal axis, we have time, the last 6 months. And on the vertical axis, we have a variant frequency between 0 and 100%. And so you can see here that at any given time, there's a small number of variants that are sort of the most predominant ones in circulation. And as time goes on, different variants sort of outcompete other variants, right? So we just see this kind of rise and fall behavior. And in the last 6 months, it's been various kinds of Omicron variants. But going back over 2 years, there's a rich sort of history of different variants coming into the picture. And so any modeling that we'd like to do should recapitulate these kind of dynamics. If we zoom in a bit, right, the previous slide was global data, but if we zoom into smaller regions like, let's say, New Jersey or Japan, then we start to see that the data are actually quite stochastic, right? So 13 million samples is a lot, but it's still, only a small fraction of the total number of people that actually got COVID. And so, there's a lot of SARS CoV-2 genomes that we never observe, right? We only observe some small fraction of what's actually going on. And that's part of the reason for this stochasticity. But also, of course, viral infection is just a stochastic process, right? Two people might be in a restaurant together for half an hour, and in some cases it'll lead to a transmission event, and in some cases it won't, right? So there's an inherent level of stochasticity in this data, right? So here again, plotted, we have something very similar to what we had on the previous slide. On the horizontal axis, we have time, and on the vertical axis, we have a fraction between 0 and 1 for a particular COVID variant, B117, and we see that, over time, the sort of broader trend is that this variant went up, and then it went back down, right? But if different regions experience different behavior, right? So for example, this variant became pretty prevalent already at the end of 2020 in, for example, California, but it didn't hit Japan until a few months later, right? So as we zoom in further and further into this data, it becomes sort of more and more stochastic. And from one time into the next, there's sort of quite a bit of stochasticity. And we need our modeling to take that kind of behavior into account. So these sort of considerations led us to develop a method that we call Bayesian viral allele selection, or BVAS for short. And at high level, this modeling and inference framework consists of 3 parts. So the first part is of data representation. So the data representation that we'll be using for our analysis is that of allele frequency space. So basically time series curves like we saw on the previous slides. So here an allele refers to a non-synonymous amino acid substitution. So for some site in the SARS CoV-2 genome, you either have the original Wuhan A amino acid, or you have a substitution to something else, right? And so we have these time series curves that encode how these allele frequencies go up and down in different regions over time, and that's the sort of fundamental data type that we'll be using to make inferences about SARS-CoV-2. So given that data representation, the second thing we need to specify is our modeling approach, and we're going to be using a diffusion-based model. In particular, we're going to do that within a probabilistic modeling framework. And the nice thing about diffusion is that it's inherently stochastic, and so that'll do a good job of being able to account for some of the stochasticity that we saw on the previous slide. And then finally, the third thing is that we'd like to explicitly assume in our analysis that most alleles are neutral, right? We're assuming that most of these mutations actually don't have any effect, or at least have a very, very small effect on differential viral fitness, right? Most of these mutations effectively don't matter, and it's only a small number of mutations that are driving the kind of differences that we see that, you know, lead to, for example, the fact that one variant is outcompeting another variant. So these last 2 points, namely the use of diffusion models and this neutrality assumption, make it perhaps interesting to reference the work of this famous Japanese population geneticist who was particularly active in the 60s and 70s, named Kimura Moto. And so he both, you know, pioneered the use of diffusion models, but he's also known for emphasizing the degree to which lots of mutations actually are neutral or approximately neutral. So there's a sense in which this framework that we're describing can be seen as a realization of some of the ideas that he's best known for. So just worth pointing that out for some additional historical context. But you know, let's unpack a bit more what this method does, right? So first of all, let's remind ourselves what diffusion is. So diffusion is something that's pretty familiar to us in our daily lives, right? So for example, whenever you make a hot cup of tea, you see diffusion in action. Here on this slide, we have a little experiment. Someone's dropping colored dye into a beaker of water. And we see that, you know, over the course of 3 still frames here, the little droplet is diffusing and expanding. So the cloud of dye expands, but it does so in a pretty complex and stochastic way. And if we were to repeat this experiment many times under identical conditions, the cloud would look a little bit different each time, right? This sort of broader pattern would be the same. The cloud diffuses outward, but the details of where those little wisps of blue go would look different each time, right? So that's the stochastic element that underlies diffusion. But there's also a deterministic component that corresponds to the drift downward, right? So in this case, you know, these glasses are on planet Earth and there's gravity and it points down. And so these clouds, despite all this sort of little turbulence and little complexities of motion that we see if we zoom in, tend to drift downwards, right? So that's what makes these kind of diffusion models interesting for us. They're a sort of principled way of combining stochastic motion with a deterministic drift component. And in this extended analogy, this drift component is going to be the genetic selection effects, right? So any allele frequency, sort of if we zoom in, might be from one time into the next, might be going up or down. But the broader trend of that allele frequency is going to be determined by some underlying deterministic drift component that corresponds to genetic selection or the fitness of any particular variant among the landscape of currently circulating variants. So let's just unpack that a bit more. We're going to describe viral infection as a diffusion process. So one of the basic concepts in epidemiology is the basic reproduction number R naught. And so what this number basically encodes is that if we have an infected individual at time t, they're going to infect a certain number of people in the next time step. And this is in general going to be a stochastic number of people. But on average, let's say that person infects 3 people, then that would mean that they have a basic reproduction number of 3. And so it's this sort of, on the other hand, there's some variance around that, some sort of distribution. And we're going to see, sometimes we'll see 4, sometimes we'll see 2. And it's being able to account for this kind of stochasticity that's going to be important in making these diffusion models sort of powerful enough to encode the actual data that we represent. And so, you know, so far I've sort of just talked about SARS-CoV-2 variants circulating in a population. But to really make this interesting, we want to connect these variants back down to the genotype, right? So this genomic surveillance data is very rich because not only do we observe, you know, a sample in a given place at a given time, but we actually know the genotype of each sample, right? Which alleles are present and which are absent, which mutations are there and which mutations are not there. And so to connect genotype to phenotype or fitness, we need to make a more detailed assumption. And we're going to make a very simple assumption, which is that the basic reproduction number R here depends on the genotype linearly, right? So we introduced this delta R term that modulates the fitness up or down. Note that in the following, I'm going to be using the words fitness and basic reproduction number and growth rate more or less interchangeably. It's just this number R, which encodes the sort of relative strength of a given variant as it outcompetes other variants that are circulating. And so this delta R term under this linearity assumption is just going to be given as a sum over capital A many alleles. In our actual analysis, we're going to have about 3,000 alleles. We introduce 3,000 selection coefficients beta, one for each amino acid. And so as a consequence, each variant its fitness, or R, is going to be governed by basically a linear combination of some small number of selection coefficients corresponding to those selection coefficients that correspond to those mutations that make up that particular variant. Okay, so just quickly wrapping up on this modeling and inference technique, the fourth word in our technique is selection, Bayesian viral allele selection. And this word choice refers to a set of techniques called Bayesian variable selection, which is just a way of imposing sparsity in our selection coefficients beta, right? So this is how our neutrality assumption sort of gets encoded at the statistical level. And so what this means is that we're going to assume that most selection coefficients beta are strictly zero. In other words, we're looking at those models which are parsimonious, and we're going to prefer solutions that are simple over solutions that are complex. And the result of doing this will be a statistical framework that tries to balance data fit, right? Trying to explain all these time series of allele frequencies, but doing so with as few non-zero betas as possible. And that's how our neutrality assumption gets encoded into our statistical framework. So before we move on to actual results, let me just leave you with one final analogy that kind of ties together how this method works. So imagine that you observe a short video of diffusion in 100 glasses of water on a 3,000 dimensional planet. So the goal in this context would be to infer the direction of gravity, right? We see 100 little clouds of ink in these 100 glasses of water, and they all look a little bit different, but all of them are tending to drift downwards. And we're given 100 such videos, and we want to infer that direction from watching those videos. And depending on how noisy those videos are, that might be hard or easy. But the point is that this sort of diffusion model gives us a principled way to sort of extract that deterministic motion and be able to look through all that stochasticity and pull out the interesting bit, which is the deterministic motion. And in this analogy, the 100 glasses of water would be the 100 regions in our analysis, and the 3,000 dimensions would be the 3,000 alleles that we're looking at in our actual genomic surveillance data. So before we move on to results from actual data, let me just briefly present you with some simulation results, right? So when you run such a complex model, it's good to see how well it does in an idealized setting with simulated data. So what I've done here is run a number of simulations where I sort of artificially create a pandemic in silico. And then I see how well our technique, BVAS, is able to recover those small number of alleles that are actually driving the dynamics in the simulated pandemic. And I'm comparing here against 3 other methods. For the details, check out the preprint. But what I'd like to sort of highlight here is a few things. So first of all, on the horizontal axis, we have depicted the total number of alleles in this each simulated pandemic. And on the vertical axis, we have some metric of success that basically encodes how many of the true alleles that are causal and that actually have fitness effects are identified by the inference technique. And so what you see here are, first of all, as the total number of alleles increases, this of course becomes a much harder problem because the hypothesis space increases and it becomes harder to identify the true alleles. But as we go from left to right and we collect more data, in this case, data across more regions, then we do a better job. And most importantly, our method, BVAS, which is denoted in blue here, sort of does the best across the board, right? So this gives us hope that when we apply this technique to real data, we actually have a good chance of extracting some of the real underlying selection effects. So now moving on to our actual data analysis from genomic surveillance data provided by GISAID. So we did an analysis on 6.9 million genomic sequences collected from 128 different regions through April 18th of this year. We looked at 2,975 non-synonymous amino acid substitutions, although you could easily look at more if you were so inclined. And recall that our method sort of delivers inferences at two levels, right? So on the one hand, we have this notion of variance, and so we get variant level inferences. For example, we get this basic reproduction number R. We're going to estimate that for each variant in our data set. But we're also going to get amino acid level inferences. To begin with, let's just look at the lineage or variant level inferences. So here I've extracted a few kind of well-known variants. On the left, we have their Pango designation. And in the second column, we have the more well-known WHO classification. And in the final column, we have the growth rate or fitness that, as estimated by our method BVAS, ranked from most fit lineages at the top to least fit lineages at the bottom. And we can see that this ordering recapitulates the kind of observed substitution dynamics that we observed in the real pandemic, right? So for example, in alpha and beta have a relatively small fitness effect over the original Wuhan A variant, whereas delta has a substantially larger growth rate advantage. And finally, some of the most recent Omicron variants have the largest growth rate advantage observed thus far. And those, of course, are the variants that have sort of dominated the recent phase of the pandemic. So this is nice to see because it recapitulates what we would have expected to see. Zooming in a bit on those results, we can also ask just what are the top 20 Pango lineages as inferred by our model. And here they are, and you can see they'll have quite large fitness advantages over the original Wuhan A variant by a factor of about 7 or 8. And all of these are various subvariants of Omicron. You know, BA5 has seen quite a bit of attention in the press recently, but these are results as of April 18th, but they are sort of broadly consistent with what we observed afterwards. So that's also good to see. Okay, so now let's quickly move on to amino acid level inferences, right? So recall that we introduced a 3,000 dimensional vector of amino acid level coefficients beta, and each of those coefficients corresponds to a specific amino acid substitution in the SARS CoV-2 genome. And so we can display them graphically as I've done here. So along the horizontal axis, we have the SARS CoV-2 genome broken up into different protein regions. And on the vertical axis, we have the inferred selection coefficient beta. So each red dot here corresponds to one selection coefficient for one specific amino acid location. And first of all, we see that most of the red dots are near zero, right? And that's as we would expect, given our neutrality assumption. And it's only a relatively small number of amino acid positions where there's a non-negligible effect size. And those effects tend to cluster in a few places. And in particular, a lot of them cluster in the spike or S protein, which is not surprising given the pivotal role that the spike protein plays in viral entry into human host cells. So this sort of recapitulates what we would have expected. And if you sort of take a closer look at this, you can see there are a few other hotspots, and some of them are in sort of parts of the genome where the biology is better characterized, and some of them are maybe a bit more mysterious or hard to understand. But all of this just points to the some of the advantages of this kind of agnostic genome-wide analysis, which is that we don't have to go in with some specific hypothesis and let's say only focus in on the S region of the genome. We can really just ask this broad question: which parts of the genome seem to be contributing to the differential viral fitness? And this can help us identify hypotheses and perhaps help prioritize experimental follow-up in the lab, and perhaps point to parts of the genome that were receiving less attention before this. Zooming in again a bit on these amino acid level inferences, here are the top 10 hits as identified by our model. And so, there are no real surprises here. These mutations that I've listed here are all kind of are mostly pretty well known and well characterized by this point. So, for example, this D641G mutation was sort of one of the early mutations that was identified towards the beginning of the pandemic as playing a role in differential fitness for some early variants. So it's good to see that kind of thing recapitulated here in our results. And an interesting thing about this Bayesian viral allele selection approach that we're using is that we, in addition to getting inferences on the actual selection coefficient beta, we also get an interpretable score called a PIP, or posterior probability, which is a score between 0 and 1 that encodes how much statistical evidence there is that supports any one of these given inferences. And so that's a useful way for us to sort of rank these different hits, but also gives us some sort of notion of how confident we should be. So these top 10 hits, for example, all have a lot of statistical evidence for them, since they have PIPs that are very close to 1, at least under our modeling assumptions. Another way that we can display our results is graphically like this. So what we have here in the left and right panel is the structure of the S protein. And in particular, the S trimer is denoted with the yellow and blue 3D mesh frame, whereas the magenta bit is the ACE2 receptor in human cells, which is sort of the main viral entry point for SARS CoV-2. And depicted in red are some of our top hits, right? These are the amino acid residues where our model thinks there's a lot of selection pressure. And if we zoom in on the right, we see that a lot of these hits are actually in this interface region between the spike trimer, in particular the so-called RBD region, or receptor binding domain, and that ACE2 receptor. So this is, what we would expect, but it's nice to see this kind of thing emerge from this kind of agnostic genome-wide analysis. Given that we have these high-dimensional inferences, both at the variant level and at the amino acid level, there are a lot of other interesting kind of visualizations and different ways that we can sort of splice and dice our inference results. And here's one of them that I like. So across the horizontal axis, we have time, basically months in the pandemic. And each blue dot here corresponds to one SARS-CoV-2 lineage or variant. And on the vertical axis, we have the delta R, the sort of fitness estimate or fitness advantage as estimated by our method, BVAS. And so, one thing that's immediately clear is that you see that as you go from left to right and as time increases, the fitness advantage of these lineages is increasing, right? So the horizontal position of each lineage or each blue dot corresponds to when it first appeared in the data, its birthday, so to speak. And we see that, a lot of the most recent lineages, of course, are the most fit, which is not surprising. But one thing that's particularly interesting in this figure is the following. So this figure, the three panels in this figure break down this delta R contribution according to the different genomic regions, right? So the leftmost figure corresponds to the estimated delta R that comes from mutations in the S gene, while the rightmost figure encodes the delta R difference that comes from everything outside of the S gene, right? So we see broadly, all three figures have the same upward trend. But there's this interesting kind of transition point at the end of 2021 where the S gene delta R contribution really jumps upward by a large amount. And that's pretty interesting because that's sort of, it's around that time period where Omicron came into the story. And so this sort of phase diagram is consistent with the idea that, ever since Omicron and ever since, late last year, we've sort of transitioned into a immune escape phase of the pandemic, where a lot of the differential fitness effects that we're seeing between different variants is driven by immune escape. And as such, mutations in the S gene are playing a particularly important role in this phase of the pandemic. So it's nice to see that in this kind of figure. So there are other kinds of analysis that you can do with this kind of setup. I'd like to just highlight one, and that's doing a vaccination rate analysis, right? So everything I described up to now is just using genomic surveillance data, but we can also try to pull in other sources of data. So in particular, one has access to vaccination rates in different regions and how that vaccination rate has gone up over time. So we can imagine a model that is similar to the model I described earlier, but now it has twice as many allele selection coefficients. We still have a vector of allele level selection coefficients beta that are going to be assumed to be vaccination independent. But now we're going to introduce 3,000 vaccination dependent effects alpha that are modulated by the time local and region local vaccination rate, which here is denoted by phi. And then we can try to infer beta and alpha from the data. And in principle, this is, this a priori, this is quite difficult, right? Because we went from inferring, say, 3,000 coefficients to 6,000. So this sort of thing is hard to do given the size of that hypothesis space. But the nice thing about the kind of Bayesian framework that we're using that also has this underlying sparsity or neutrality assumption is that it, this sort of thing still remains tractable. And we're still going to, use our parsimony assumption to favor simple solutions. And the strongest evidence that emerges from this kind of analysis for allele level dependence on vaccination rates is in an interesting residue in the spike protein called N501. And so it appears from our analysis that the vaccination actually confers differential protection against this allele, right? So we know that vaccinations confer protection against SARS-CoV-2 variants in general, but this analysis suggests that there's sort of an additional differential protection against those variants that carry this particular allele. So I'd like to quickly summarize. So at the beginning of the talk, we asked these 5 high-level questions. And so let's sort of see how we did. Which of these questions were we able to answer with our method, BVAS? And so I would argue that the first 3 questions, we did a pretty good job. So I put a red check mark. So those questions are: which mutations are driving the pandemic? Which variants are the most fit? We get a nice PIP ranked list of mutations. So that's useful for prioritizing experiments, for example. We also get, we have the ability to make fitness estimates for newly emergent or putative variants that maybe don't exist in the data at all or have only barely been seen. And that's relying on the statistical sharing of strength that we get by our linear fitness model. So that's useful. The next question was, which mutations are implicated in immune escape? With this analysis that I described on the last slide, this vaccination-dependent analysis, we sort of get an initial attempt of an answer at that. I've only put a sort of orange check mark here to note that this is a particularly difficult question to answer using genomic surveillance data, right? Because the vaccination data that we have is at the population level, right? Whereas what we'd really like is to have vaccination data at the patient level, and we would like for the patient data to then be connected to the genomic samples, and then it would be, in principle, easier to make, to do this kind of analysis. Nevertheless, we were able to extract some insights using this population level data, so that's interesting. And finally, another interesting question is there evidence for epistasis? So I didn't go over this in the previous slides, but if you're interested in this, check out the preprint. An interesting thing about epistasis from our point of view is, there are many mutations observed in SARS-CoV-2, so it becomes a priori difficult to answer questions about epistasis, since epistasis is a question about how different mutations interact. And if you have thousands of mutations, then you have millions of possible interaction effects. But one nice thing about our framework is that because we have this sort of Bayesian sparsity assumption at the core of our model, it's actually tractable to try to answer some of these questions. And so some epistasis effects may, in fact, you may be able to extract certain epistasis effects from this kind of data, although doing so is quite hard for a number of reasons, including the fact that most combinations of alleles are just not present in the data, right? We've seen thousands of variants, but we haven't seen the millions of possible variants that one could have, right? So it's still difficult to do this, but there is some tantalizing evidence of epistasis in the genomic surveillance data that we've analyzed. So if you're interested in that, check that out. And more broadly, if you're interested in our work, check out our preprint. There's also an open source library for BVAS on GitHub, which is available to anyone who's interested. And in general, please feel free to ask me any questions. And thank you so much for your time.